Sir,
Madam, yeah,
we mainly have life science field. We have faculty members and research scholars
okay.
with us from
From
various life
different,
science field.
right, right. Okay, okay, great. And I have about 1 hour to,
Yes, sir.
or
Sure, sir.
there will
You
be
have
question
about 1 hour.
answer. Okay,
Yes, we can go on for 1 hour and we can take up question answers for the next 10-15 minutes.
okay. Thank you. So I will be sharing my screen. Just a moment. Can you see my screen?
Yes, sir.
Okay, great. So I will be.
But sir, it is not in the full screen mode.
Okay. Yeah, now?
Yes, now it is perfect, sir.
Great. So thank you for the kind introduction. Actually, my area of research presently is hovering around maybe polyextremophilic organisms from different sites. And that actually I started in the year 2019 onwards. And this particular area, apart from this area, I am also doing some work on secondary metabolites from plant sources. I'm also trying to do some work on bioaerosols, but they are all under environmental microbiology. So basically, you can call me a person who is very much close to environment and agricultural microbiology. So when I thought of delivering this particular lecture actually is not only having agricultural, but I wanted to give a touch on certain industrial importance of the secondary metabolite from these microbes. So now, polyextremophile, the journey from extremophily to polyextremophily is actually very interesting because the organisms that we collected, that was that actually I collected from at a quite a high altitude of 3,500 meter, and that is basically in Gangotri, next to the Gangotri Ghat, and also Jamunotri, the hot water spring, which actually is having a high TDS content, which is quite obvious. But very strangely, the pH is slightly alkaline. The dissolved oxygen is actually on the lower side and quite low, in fact, which actually makes it suitable for microaerophilic organisms. And the salinity was also on the lower side. But why I actually collected these organisms? Not for pilgrimage purpose. I just visited there as a tourist and I collected in some sterile container. I had this objective in mind that what type of bacteria we normally get in that higher stage. And this particular work, because it was so nice that it resulted in 1 or 2 PhD. So obviously within the span of 6 years. So in my next outing in Ladakh, I did the same thing, collected water sample from Somoridi and Pangong Lake. But I could find that result was not that good as I actually got it from Gangotri and Jamunotri. So, of course, the samples were brought and in the laboratory, we isolated the organisms. But before going into these organisms, I just wanted to share some of the information which were actually, these are hand-drawn sketches by one of my scholars. What actually this particular polyextremophile mean? They may be saline tolerant, they may be barophiles, they may be metallophile, that is tolerant to different heavy metals. At the same time, they may produce different types of industrially important enzymes. And obviously, these enzymes can actually be of maximum use when grown in relatively higher temperature or pressure, and which makes them a very important source in terms of industrial compounds. At the same time, they do have agricultural importance in three different aspects. One is they are actually recycling complex matter, organic matter. They are also antioxidants, and they are also producing some volatile substances which gives an all-round protection. Finally, they have the ability of common PGB, that is nitrogen absorption, phosphate, and potassium solubilizing activity. Apart from that, as I was telling, most of them, they have a unique ability to produce extracellular polysaccharide, which enable them to do the Coram sensing, and they do have prolonged CAMP activities. They do have diGMP. These are very common parameters which are found in this particular bacteria. So when I will be discussing their genome pattern, I'll be touching upon this particular aspects. So I have structured my lecture in such a way, I'll be talking little bit about these organisms, then I'll be doing little bit on the whole genome, and partially I'll be mentioning some of these genes and their importance. And then I'll be getting into some of this ESI-MS data, which will show low molecular weight metabolites, which indicate that they are not only important in the agricultural field, but they are also important in industrial arena. So as I was telling, that these PGP activities, which we found out, that was basically in the form of, just one moment. হ্যালো, হ্যাঁ তুমি তো 4টে বললে আসবে। আচ্ছা তুমি এসো, তাহলে এসে কন্ট্রোলারে এসে টক টক করো, আমি বলে দিই, মুখটা বাড়াবে আমি দিয়ে দেবো। Now, as I was telling, these PGP activities include not only nutrient procurement, but also some sort of drought tolerance, saline tolerance, heavy metal binding through the production of not only siderophore, siderophore is only for iron, but also through the production of metallothionein. And sometimes these metals may also include zinc solubilization, which is good for the plant growth. And they are producing biofilms, which are actually very common for these bacteria. And these biofilms not only for the pathogenic activity, but here it is for the importance of the plant growth. Now, when I was doing this particular Firmicute biocontrol activities, many of these compounds we actually found out. That included siderophore, lipopeptides, antibiotics, and they are capable of producing induced systemic resistance in plant. And that is actually a very good point that we normally see in case of a normal PGB. So the bacteria that we isolated, actually we isolated about 12 on the basis of the common growth. We never did the metagenome, but we straight away went into wet lab. We never started with whole genome analysis. We started with 16S identification. And then we actually wanted to see them under electron microscope. And accordingly, we reported their sequences in NCBI and we gave a unique name BRAM G1, G2, G3, Y2 and Y3. We had Y1, but ultimately we couldn't protect this bacteria. It didn't grow under normal condition here. But the remaining 5, they grew. So the first one, which we had it, was Bacillus subtilis. And this was reported as BRAM G1. And we see this approximately 9 megabase gene. And here in the gene sequence, if you can see that many of these genes, apart from normal housekeeping ones, they have some unique property which enable them to survive in different stressful conditions and also minimize the competition. These stressful conditions may include absence of oxygen, because you see, I've seen that dissolved oxygen was pretty less. Production of some secondary metabolite, which will be acting as antibiotics for others, having their own basic antibiotic protection, producing some antibacteriocin properties. And these antibacteriocin properties are unique through the expression of RCS or LGR gene. Apart from that, they do have this unique secondary metabolites like octopine production. They have methamphetamine, which actually can reduce biofilm, but very strangely, their own biofilm, when they are producing, this particular gene was not functional. Apart from that, they did have natural UV protection mechanism, production of pullulanase, which is in the production of biofuel. And actually speaking, when we did the whole genome of all these bacteria, we found out that they are good as a player. I will be showing you the results, but individualistic treatment may be even better. So some of the players are really very good players and they do not give their fullest performance when grown in group, but they give very good result when actually applied in single or in double. So this particular bacteria was definitely a strain of Bacillus subtilis, which you have already reported as a novel strain. And we have actually maintained since then in the laboratory. Mind you, these bacteria, as on date, still they have retained. We have been doing, after even the PhD of Bedoprana, we are still continuing our work, and they have retained their polyextremophilic property. That is, the journey from extremophily to polyextremophily, as on date, holds good for this particular bacteria. The next one was actually a unique pest killing bacteria of Bacillus thuringiensis. And this was also reported as BRAM G2 from Gongo 3. And apart from the normal DNA, RNA replication, maintenance, transcription, translational genes, they have lot of separate genes. And some of these genes are very common. They're very common. That is, হ্যাঁ তুমি দাঁড়াও আমি দিচ্ছি। এই অজয়, বাইরে একটা ছেলে দাঁড়িয়ে আছে, ওকে ভেতরে ডাক, একটু মুখটা বাড়াতে বল। Now, we have, apart from the normal genes, that is Bacillobactin, siderophore, or presence of pullulanase, UV protection, surfactant, these were already there in the previous case. Apart from that, some very interesting genes were there, which included production of some unique EPS compounds, additional mechanism for histidine utilization, sugar breakdown from glycogen, that is gluconeogenesis, not normal sugar, transferring the amino group or removing the amino group for the interplay between keto acid and amino acid, UV repairing mechanism, nitrate reductase mechanism, which are again very common, but very interestingly, they have some very interesting chemotoxic protein, which is accepting the methyl group, methylation of chemotoxic protein. They have the ability to detox nitric oxide and protect themselves. They have the ability to produce antioxidants, not normal this oxidase or other enzymes, but they have the ability to produce hypoxanthine. They do have calcium transport mechanism, which is also unique for the rigidity of the cell wall of this particular bacteria. So this was labeled as G2. The third one under this list was BRAM G3, which was identified to be Bacillus teculiensis after whole genome analysis. And each of these bacteria, they were all producing biofilms. But we could see that they had some interesting genes. If we consider the stresses, the primary stress comes from starvation, from other metabolites, UV damage, and for which they have stress regulating genes or polyketide producing gene or nucleotide excision repairing genes. Production of nitric oxide metabolizing enzyme, production of bacillicin, surfactant, and other antibiotics, production of very common glycogen metabolizing enzyme, the chemotaxis enzymes. But what is interesting, we got some policy which actually is triggering immune response for future protection, which is another interesting point that we got from Bacillus teculiensis G3. So you can very well understand these three bacteria, that is G1, G2, and G3, they were actually different. You will say, how different? Well, Bacillus thuringiensis, Bacillus subtilis, they are not different, but they are different in their activity. the name may be the same, but they have developed certain unique polyextremophilic genes or switched on certain genes that have made them capable of surviving. ওকে বলো ওই অভিজিতের কাছে ওর রেজাল্টটা ছেড়েছে কিনা একটু যেন ওটা কাগজে লিখে দাও। না না আরেকটা ওটা কাগজে লিখে দাও, দিয়ে অভিজিৎকে দাও, ওটা ছেড়ে দিতে বলো। ওকে আচ্ছা এক কাজ করো, ওকে তুমি অফিসে আগে অ্যাপ্লিকেশন দাও, অফিসে ভাইস প্রিন্সিপালের কাছে, যাও। না পরে, এখন ক্লাস করছি। Now, so G1, G2, G3 were important in their own way, but they were different from the normal strains, and tequilensis as such, I could say that they are different because they had some unique regulating genes that made them survive in unfavorable condition. But very strangely, this particular bacteria in common, they all produced biofilm. They were all capable of generating extracellular polysaccharide, which could enable them to withstand both low and high temperature. And you must understand that these bacteria, they are growing at that point where the temperature can go down to -10, -12, -13 during winter condition. At the same time, this particular bacteria, which we obtained from Yamunotri, Surjakund, they were capable of withstanding high temperature because they were from a natural sulfur hot spring, which is not getting frozen during winter. And this particular bacteria were reported as Y2 and Y3. As I told you, Y1 was not there, or rather it was there, but we couldn't maintain it. So this particular bacteria incidentally was considered to be bacillus when we did the 16S rRNA, but later on, doing the whole genome, we found out that this bacteria was Mesobacillus thioparens, BRAM-Y2, as we reported it to NCBI. Here, some unique genes that we got, that was, one was definitely anaerobic or nitrogen metabolizing Gogurt gene, tetrathiamide gene. Yes.
Arup sir, sir, are you on this slide that BRAM-Y2 to be a unique strain? Or
Yeah.
are you changing the slide? Okay, somebody was asking that are you on a different slide or you're explaining?
No. BRAM-Y2 only.
Okay.
I'm
Thank you, sir.
considering certain genes.
Okay, sir.
This particular bacteria, they had, in fact, this is true for Y3 as well, certain genes which I actually found out they are expressing in fungal members like in yeast. So that is a different story altogether, where there is evolution of some particular genes to higher order organism. And so there will be some phage transcript genes which also has originated due to specialized transduction, which I believe is a very common phenomenon. Apart from that, we have seen certain unique recombinase gene, RECI, which actually was bringing about the normal recombination and also DNA repair or DNA damage repair. There are some unique COMIC gene with which they can acquire or procure DNA from the nature, but yet they maintain their own rigidity. They were copper efflux gene, and mind you, in certain bacteria, I have not marked always, they are producing multiple export genes, which actually indicate that under severe heavy metal stress, more than one efflux pump will work and they will pump out this particular copper toxicity. Of course, here I am not considering the normal clamp, DNA repair, gene recognition. All these genes are very common. But overall, the export of guanidium or production of mycothiol indicate that certain genes, the story doesn't end with bacteria. It can actually got into some other fungal members and reported from other members of eukaryote. Now I have changed the slide, that is BRAM Y3. Is it visible?
Yes, sir.
Yeah. So this particular bacteria, I will later on showing in some of the results, is unique because it is actually protecting from different types of secondary infectious organisms growing in croplands. So an organism which was isolated at an altitude of around 3,500 meter is equally functional when they are grown in the plain, and that also in a consortia. Of course, after doing the t-test, they played a beautiful role in agricultural productivity. Now here also, I will not mention the one which I have already stated, but apart from that, they have the ability to produce vitamins, which is unique. I will later on come to that. Vitamin K, production of small peptides, and these small peptides can be replacing antibiotics for other particular organisms. They produce unique sensor kinase enzyme, which actually will activate the different cell divisional machinery when they sense some sort of stress in the vicinity. They have the unique cyclophilin for mitogen activity. They have the unique DNA repairing enzymes. They have the tyrosidine and gramicidine, which are secondary metabolites for other organisms. And they not only produce the oligopeptides, but also help in their transport outside, which actually will reduce the competition when grown in a mixed condition. So this particular bacteria was unique for us, and it is still playing a very gigantic role in our further research as Brevibacillus parabrevis. As I was telling you, single stress, well, growing in UV, growing at high temperature will just prove its extremophilic property. But we are not happy with it. We were putting them in nine different three-dimensional stresses where we have actually made them grow in high temperature, pH, salt, any three at a time, or sometimes PEG, that is polyethylene glycol, to indicate typical drought condition. We also actually tried to use them or grow them in UV. Apart from UV salt, drought. In this way, we made combinations. And you can see some of these organisms, like I would say all of them, but to some extent the Y3, in some cases the G2, they were showing excellent result when grown under 3D stress. Nutrient acquisition, you can see that G3, Y3, they are doing a good job, particularly with respect to zinc, or not zinc, basically potassium and nitrogen fixation, potassium solubilization, nitrogen fixation. Though we didn't get phosphate solubilization at the outset. Production of auxin and gibberellin were more or less carried out by all of them, but some of them actually working better in presence of tryptophan. And here also I have compared it with normal bacillus, where auxin production is higher in comparison to normal laboratory strain bacillus. Now this is a very important enzyme. What is that? SEC deaminase. This particular SEC deaminase, as you know, is very important because it will protect the organism from normal ethylene stress. And you know, plant, they do produce ethylene. So if SEC deaminase is there, that will reduce the stress of ethylene. They had the ability to produce siderophore, as I have already stated, because I have marked the siderophore genes. So it is quite obvious that they will all produce siderophores. But what is interesting, that they are producing mainly two types of siderophore, that is hydroxymate and catecholate type. Well, you all have seen that oxygen stress, in order to remove that, they can grow by producing catalase, which is very common for any bacteria, or peroxidase. But at a very high level, they are producing this. What is important, the vertical bond, that is beta-1,3 glucanase, is being produced by this bacteria, particularly by G3 and Y2 and G1, which actually make them potent antifungal bacteria. More or less, in comparison to normal bacillus, all of them, they could degrade biomass, increase the biomass through the degradation of pectin, through the degradation of plant secondary wall substances, lactase, and also cellulase. And these are really very important in terms of agriculture, because where you don't get good soil, but to create that soil, you need to degrade that particular biomass, and that is being ideally done by this particular bacteria. So after testing their ability to help in agriculture, to degrade biomass, to produce phosphate solubilizing, or here it is potassium and nitrogen fixing solubilizing activity, we use them in the growth of maize plant. And actually we could see that in comparison to consortia, the growth in case of Y2 and Y3, it was really very long. And here it is comparable to that particular long plant, which is a very common, disease that we see in the form of Bakane disease, brought about by Gibberella fujikori. So it is not a disease though, but because of high gibberellin production, the internode length has gone up and the plant height has increased. That we found out particularly true for Y2 and Y3. Mind you, when we actually prepared this particular consortia, we all did the interaction study so that safely both can be applied together. Then we actually started to use them in paddy, basically. That was against some abiotic stresses. What type of stress? Arsenic, which is a very common stress, salt stress and drought stress. Now, this particular treatment, arsenic can be directly or salt directly can be applied to the soil. And for drought condition, we initially grow the seedling under low moisture by absorbing the moisture and putting them under drought stress and then exposing them to normal growth condition. What we observed was that this particular plant, more or less in comparison to control, all the setup, they showed normal growth when the PGP treatment was given. And here, actually, all the arrangements, they grew well. And here we used all the 5 consortia together, not Y2 and Y3, unlike the previous case, just to see that whether they are making the plant stress resistant. Except for drought stress, we see that particularly arsenic, salt, they were growing very well. Though these are short-term tests where we used all the 5 bacteria together under different types of stress, and we didn't use multiple stress at the same time, but individual stress for individual setup, and the result was really good. I have not included their individual chlorophyll or individual internode length or individual number of leaves, primary productivity, because it is obvious from the growth, at least in the PGP treated sets, the plants were not that much inferior with respect to control in a 6-week study. Of course, you will say, why 6 weeks? Because after 6 weeks, more or less, the plant becomes quite accustomed to the environmental conditions. So up till now, what is the take-home message? The take-home message is these extremophilic organisms can actually modify the soil and make them grow well or make the plant grow well. From the hills or from the mountains of Himalayas to the plains, they have retained their PGP activity, and which actually give them a very good property of all season fertilizer mixture. We didn't stop here. We did the ESIMS, and the ESIMS data actually was showing different typical peaks. Most of them were producing some antibiotics. Obviously, for Bacillus, bacitracin is very common. But one thing you can see, they are producing surfactant. Incidentally, I have shown you in some previous slide that though surfactant is being produced, but surfactant is not causing toxicity because they have the ability to protect themselves from surfactant. And in addition, they also produced, all of them produced niacin. The SMI's data actually showed that, and this actually is produced. You can see the lower one is controlled under the stress, under the 3D stress. When these organisms were gone under the 3D stress, these secondary metabolites are being produced. It's not that they are producing because of this temperature UV, but because of this drought or because of this UV and because of high temperature, the secondary metabolite in the form of antibiotics are being generated. So this particular slide will give you a better idea. I have also highlighted some of them. which you can understand that they are producing biotin, which is a vitamin. They are producing anticapsin. All the strains, the names are given there, which is actually making them resistant to bacillicin. They are producing levanase like G2, which is producing levan oligosaccharide production. That is alternative sugar breakdown. They are producing antibiotics like azoxyvacillin, and a very interesting one, they produce a linear lipopeptide, which is also antibacterial in nature in the form of gagiotetrine. I have shown you already the peaks were there. They were producing nishin, which is actually antibacterial, but for increasing the shelf life of different food material. They were producing pyrazine. Pyrazine is actually a scaffold which is having antimicrobial or anti-inflammatory activity, which is good as a medicine. They produce metalloprotease. They produce fungicin, which is also a fungicide antifungal compound. They were producing bacillin. Bacillin is actually an antibiotic again, and surfactant already I have stated because that was there in the peak, quite evident. Very interesting, you see. They were not liberating in the surrounding, but they were also keeping them in their biofilm, biofilm retention. And that include different secondary metabolite like hexatricontin or ligoglucamide, all antibiotic compounds. They were producing vitamins like cyanoglucobalamine, vitamin B12. They were producing 2,4-dimethyl phenolic compounds, which are antifungal, preventing the growth. And they were quite prominent. The peaks are quite prominent. This is G1, this is G2. We come to G3, where again this trend is there. Apart from that, there may be argotaman. Argotaman is actually a particular alkaloid which is producing argotamine. What is that? Which is inducing, the smooth muscle contraction causing abortion. They were producing essential fatty acid like octadecatrienoate. They were producing pyrazine, bacillin. All these things were there. And you can see the frequency of the peak in Y2 is more. So secondary metabolite production may be more in Y2 and even more in Y3, like leodoglucamide, adenosyl cyanocobalamin, and others. Some of these compounds have been clearly given here. Some common ones include vitamin B5, vitamin B1, vitamin B9. So just see, they actually can be a commercial source of vitamins growing in the Himalayas, can be brought to industry producing these compounds. They include the canosamines, they include violacin, it's a very important compound, particularly as an anti-cancer drug. It is also producing bioplastic. It is producing iturin from G2, a non-ribosomal cyclic lipopeptide, which is actually having antibacterial and anti-cancer activities. Argotaman, apart from smooth muscle contraction, is also used in the treatment of Parkinsonism. So these are host of compounds that we see, which are actually deposited in the biofilm, and we have got this after we did the SIMS of the biofilm only and not the bacterial cell. What does it mean? It means that these secondary metabolites are coming out of the cell, they are deposited in the biofilm and can be easily harnessed, which is making them a very important industrial compounds. So you can see the major conclusion from this particular lecture was these particular strains which we obtained from the Himalayas, the Bacillus subtilis, Bacillus teculiensis, Bacillus thuringiensis, then the Mesobacillus thioparens or Brevibacillus parabrevis, they were all having polyextremophilic property. They were having PGP activities, and these PGP activities are not only helped in plant growth but also protected them from different stress like ethylene, from fungal infection, and even they helped in the growth of the internode, making them very well suited to this particular environmental condition. They were even helping them to survive under arsenic stress. That was the agricultural part. But under 3D stress, they were not only stable, but they produced different types of secondary metabolite, which actually has resulted in different compounds. And if they can be purified, they will produce these anti-cancer, antibacterial compounds in bulk. But the problem is, we are here not for industrial production. The reporting was done, papers were published, but future condition of growing them industrially lies with the hand of the industries. Now, this resulted in some papers and book chapters. So this was one of them from an Elsevier book, Unique Extremophilic Bacillus, their application in plant growth, promotion and sustainable agriculture. This is another one, high altitude biofilm forming novel Bacillus subtilis, that we did this work in ZMAs, and this was published in Applied Biochemistry and Biotechnology. This was a paper. This is also another paper. We did the biofilm forming Bacillus valismotis, the T rhizosphere. This was another interesting bacteria that we obtained. I didn't show it here, but this was also a very unique one, which is growing in the T rhizosphere. So that is all. Thank you very much for your patient hearing. Thanks to the organizer for this invitation. Thanks to Dr. Prasun Trivedi for inviting me. Thanks to our principal, our head of the department, and of course, the work which I have presented here is because of the hard work of my scholars like Veduprana, Devopriya, Vidisha, Sanjana, Vikram, all of them. They are actually helping me to do some work in environmental microbiology, which I have shared with you. So thank you very much. If there are any questions, I am free to answer.
Thank you, sir. You have discussed on a very core microbiology topic, and I'm sure that our audience and participants have gained a wealth of knowledge from this lecture. So thank you, sir. And I'm looking forward to any questions from the audience. Any questions?
Hey, second year
Actually, I'm sorry because I have to do little bit administrative work. So there has been some disturbances while I was delivering the lecture. Please excuse me, pardon me for this.
No issue, sir. It is fine. Sir, I think there are no questions from our audience.
That means
thank
two
you
things.
once
Either
again.
they have understood everything or they have not understood anything. Anyway. But this, let me tell you this entire work which I have presented our own work. I have not taken anybody's work, not nor did I present any review. I actually wanted to tell you that we have treasures in our own country. If we can harness it properly, that can actually give us more mileage in future.
Okay, sir, there is a comment in our chat box. It is from
Yeah.
Vinith Moria. He's saying that he has no doubt. It is really, it was a nice work focused on extremophiles from Himalaya. So he really liked your presentation. That is what he said.
Thank you. Thank you. Thank you
I
very
have a
much.
And
comment.
okay, we have some doubt, I think. Yes, please.
Ma'am, I am Dr. Ishita Rahman. So, sir, I just want to comment. The lecture was very informative, and we gained much knowledge after hearing the lecture. Sir, one question, sir. Actually, in our class 12, I read your book. I mean, your life science book also.
Okay.
You have the book, right? The
Yeah.
class 11,
Yeah.
12. So I mean it. I mean I followed that book for my board exams, and I mean it's a wonderful thing that I have an opportunity to interact with you in this forum. And thank you, sir. The book was very well written. I
Thank you.
followed that book. Mhm. Thank you, sir.
Thank you. Thank you. Thank you.
Thank you, Dr. Rahman. So I think, sir, we are getting a lot of messages on that. It was a very informative and well
Okay.
explained
Thank you
presentation.
very much. Thank you. It was
Thank
my
you,
pleasure.
sir, once again for taking
Okay.
out time from your busy schedule. Thank you, sir.
Okay. I take leave. Thank you very
Thank
much.
you. Okay, participants, please follow the group. You will be shared a link, which is the assessment link in the FDP group. Please fill that up. Okay. Try to finish it within half an hour. And those who cannot, please try to fill it up by today itself. Okay. After which you will not be allowed. Only if you have any problem, you can delay the time, but it is preferred that you finish it by within this half an hour. Okay. So I will be sharing the link in the group. And thereafter, again, we will join. And